Bismillahirrahmanirrahim. Assalamu alaikum dear student. Welcome to your channel The Anatomy Canvas with your tutor Dr. Shehla Aman. In today's class of embryology of the nervous system, we are going to discuss with the development of the hind brain and the midbrain and discuss the development of the alar and basal plates and their derivatives in the area of the hindbrain and the midbrain. So first have a look on development of the brain. Cranial to the fourth pair of the somoid neural tube develops into brain. Fusion of the neural tube in cranial regions form initially three primary brain vesicles. These vesicles are called forebrain, which is called prosencephalon, the midbrain, which is called mesencephalon, and hindbrain, that is called the rhombencephalon. During further development, forebrain divides into telencephalon and diencephalon, while hindbrain divides into the metencephalon and myelencephalon. The midbrain does not divide, so there are the five secondary brain vesicles. Initially, we have three brain vesicles, and later with development, we have the five secondary brain vesicles. So this is the diagram. This is the initial one in which you can see the three vesicles, the three primary vesicles. This one is the forebrain or the prosencephalon, this one is midbrain or mesencephalon, and this one is the hindbrain or the rhombencephalon. Later with development, the prosencephalon divides into telencephalon and diencephalon. Mesencephalon remains as such, and the hindbrain or the rhombencephalon then divides into metencephalon and myelencephalon. So telencephalon will form the cerebral hemisphere, and the cavity of the telencephalon is called the lateral ventricle. The diencephalon will form the thalamus, the hypothalamus, etc., and their cavity is called the third ventricle. Midbrain remains as such, the mesencephalon forming midbrain and its cavity is called aqueduct, then metencephalon forming pons and cerebellum and forming the upper part of the fourth ventricle, while myelencephalon forming the medulla and it is forming the lower part of the fourth ventricle, and from here the spinal cord starts, whose cavity is called the central canal. So these are the initial three primary brain vesicles and five secondary vesicles and their derivatives and their cavities. This is the diagram. You can see this is the area of the forebrain or the prosencephalon. Here you can see the optic vesicle is developing. This is the area of the midbrain and this is the area of the hindbrain. So a flexure is produced in between the midbrain and the forebrain because of the growth and because of the folding of the forebrain. This is called the midbrain flexure. Another flexure develops over here and this is called the cervical flexure. Later on, a third ventral flexure also form called the pontine flexure. So we have initially during development the embryonic brain grows rapidly and bends ventrally with the head fold. So in this diagram you can see the bending and the folding of the embryonic forebrain, and as a result of the bending and folding a flexure is produced which is called midbrain flexure, also called the mesencephalic flexure. Also at the junction of the myelencephalon and the spinal cord, because of the differential growth rate, another flexure forms, and this is called the cervical flexure. So later the unequal growth of the brain between the flexures produces the pontine flexure, and this flexure results in thinning of the roof of the hindbrain. So in this area you can see on the back area you can see the pontine flexure will form over here. So cervical flexure demarcates the hindbrain from the spinal cord, and later this junction will be defined as level of the superior rootlets of the cervical nerves. The pontine flexure divides the hindbrain into the myelencephalon, that is the caudal, forming the medulla oblongata, and metencephalon, which is cranial towards the front, develop into the pons and the cerebellum. So these are the two flexures. The cavity of the hindbrain becomes the fourth ventricle and the central canal in caudal part of the medulla. So regional expansion, the prosencephalon, this is the area of the prosencephalon, this is the area of the mesencephalon, and this whole is the area of the hindbrain or the rhombencephalon, and below is the spinal cord. So these different colors shows the developmentally different areas. So this is a prosencephalon forming the telencephalon over here, and these two areas diencephalon 1 and 2. This is the mesencephalon, and these areas are the rhombencephalon, and here is the spinal cord. So here you can see three flexures which are forming. Two are concave ventrally and one concave dorsally. So here is the mesencephalic flexure which is present ventrally, and the cervical or cervical flexure which is present also ventrally, and the one which is present posteriorly or dorsally over here, this is the pontine flexure, which divides the upper part of the hindbrain from the lower part of the hindbrain. So because of the changes in the cranial part of the medulla oblongata, there is the thinning of the roof plate, and as a result of thinning of the roof plate, the changes occur and the fourth ventricle will be formed. Also you can see the in the area of the alar plates, the neurons migrate from the area of the alar plates to the marginal zone, and here they form the gracile nucleus and the cuneate nucleus. In the anterior part you can see the pyramids are forming, which are composed of the corticospinal fibers. Here you can see in this diagram, this is the basal plate which is forming different nuclei, and this is the alar plate. So neuroplasts from the alar plates in mesencephalon migrate into marginal zone and form isolated areas of the gray matter, gracile nuclei, and the cuneate. So in this diagram you can see from the area of the mesencephalon, in the area of the mesencephalon, neuroplasts are migrating from alar plates, and they are forming the gracile nucleus which is medial one, and the cuneate nucleus which is lateral one. The ventral area of the medulla contains pyramid, a pair of the fiber bundle. So here you can see the pyramids. During further development, as walls of medulla move laterally, the alar plates lie lateral to the basal plates. So motor nuclei medial to the sensory nuclei. So in this diagram, you can see the lateral plates of the medulla are moving laterally. Walls of the medulla are moving laterally, like opening of the page of the book, as a result of which the alar plates lies lateral to the basal plates. So these are the basal plates and these are the alar plates, and basal plates forming the motor nuclei and alar plates forming the sensory nuclei. So mesencephalon to the pontine flexure causes lateral walls of the medulla to move laterally like pages of the book. So in this diagram you can see these are thinned out area in the area of the pontine flexure, and these are the changes which are present in these areas. So now we move on to different types of the nuclei which are formed from the different cells. First we will discuss the nuclei which are forming from the basal plates, which is basically the motor area. So in this diagram you can see the most medial one. The most medial nuclei is the general somatic efferent. Means these are the neurons of the hypoglossal nerve in the area of the mesencephalon, and these are carrying the general somatic efferent, means general motor fibers which are carried through the hypoglossal nerve. Next to the general somatic efferent is the special visceral efferent group, and this special visceral efferent groups neurons innervate muscle derived from the areas of the pharyngeal arches. Next to this is the general visceral efferent, and this general visceral efferent carries neurons of the vagus and glossopharyngeal nerves. These are carrying the motor fibers to the viscera like intestine and the cardiovascular system, the respiratory systems, etc. And Next, we are discussing the Eller plates, the neuroblasts in the Eller plates which are forming different motor nuclei. So please remember, in the basal plates we have 3 sets of the nuclei, while in Eller plates we have 4 sets of the nuclei. So next to the next to the general visceral efferent is the general visceral afferent group. This general visceral efferent receives impulses, sensory impulses from the viscera. Next to general visceral efferent group is the special visceral efferent group, which is carrying the taste sensation, and after this we have general somatic efferent, which receives impulses from the surface of the head, that is the general sensations of the pain, touch and temperature, etc. And the last one is the special somatic efferent group, which are carrying impulses from the ear, and also the olivary nuclei are also formed in this area from the Eller plate. So this organization is very important. You have to memorize, because all of this will be visible in not only in the myelencephalon but in the metencephalon as well as in the mesencephalon, and also in different areas. So these nuclear groups are present, which will give rise to different nuclei for the cranial nerves. So again I repeat, first the most medial is the general somatic efferent, then is the special visceral efferent, then is general visceral efferent. Next to general visceral efferent is the general visceral efferent group, and after to that is the special visceral efferent group, then general somatic efferent and special somatic efferent. So this is the diagram from it, cross section, longitudinal section from the adult brain, just to have a look and to understand the different nuclei and their derivatives in the adult brain. So first, this color show the somatic, general somatic motor or general somatic efferent fibers. So these general somatic efferent fibers in the region of the mesencephalon, they are shown by the oculomotor nerve and the oculomotor enterotrochlear nerve, and in the region of the pons they are shown by the abducent nerve nuclei, and here they are in the area of the medulla oblongata, they are shown by the hypoglossal nucleus. So these 4 nuclei, these are derived from the general somatic efferent group, the oculomotor nerve, the trochlear nerve and the abducent nerve and the hypoglossal nucleus. This is the first class, remember, the most medial one. Next one, the fibers which are supplying the brachial motor fibers to the brachial arches, which are the general visceral efferent fibers. And these are special visceral efferent fibers, and these special visceral efferent are shown here by the these are the fibers which are shown over here by the trigeminal motor nerve nucleus, and then the facial motor nerve nucleus, and then the abducent nucleus, nucleus ambiguous, and then the accessory nucleus. So these 4 nuclei, the trigeminal motor in the area of the pons, then the facial motor in the area of the pons, then the dorsal motor, this is nucleus ambiguous, which are carrying fibers both from 9th and 10th nerve, and then the accessory nucleus. These are all the fibers which are supplying the pharyngeal arches. Next to this is the general visceral motor fibers, and these are shown by over here in the region of the mesencephalon. These are shown by the Ettinger-Westphal nucleus, which is supplying the pupillary sphincter, pupillary muscle of the eye, and then over here it is shown by the dorsal motor nucleus of the vagus. So please memorize these nuclei, these are very important. And in addition to this, the 2 smaller nuclei, the salivatory nucleus, also over here. Then we move on to the sensory nucleus. The general sensory nucleus shown in light yellow color. This is the mesencephalic part of the trigeminal nucleus, the principal part of the trigeminal nucleus, and then the spinal trigeminal nucleus. So this is the general sensory or general somatic efferent fibers. Then is the visceral sensory fibers, which are shown over here by the nucleus of the tractus solitarius. and then the special sensory, which are shown over here by the cochlear nucleus and the vestibular nuclear complex. So these are the different nuclei and their derivatives, which are initially formed from the alar plates and the basal plates, and their derivatives in the mesencephalon, in the bones, and in the medulla oblongata. So please memorize. This will also help you to memorize different types of the fibers in neuroanatomy as well. So have a look at this table just to memorize. In the area of the mesencephalon, the general somatic efferent fibers are shown by the oculomotor nerve, the trochlear nerve, and the abducent nerve. These are supplying the striated muscle fibers to the extraocular muscles of the eye. Then the special visceral efferent fibers, which are carrying the supply efferent fibers motor sensation from the viscera, that is the pharynx, the striated muscles of the pharynx. This is cranial nerve 5 and 7. Then the somatic efferent fibers in the area of the mesencephalon. These are the general sensations of pain, touch, and these are the sensation of the taste. These are the special efferent carried through the cranial nerve 7 and 9. Then the special efferent carries through the cranial nerve 7, having balance and hearing, and then general somatic efferent, which are having that sensations of the pain, touch, temperature from head and neck region through cranial nerve 5, 7, and 9. So these are the different nerve nuclei which are present in the area of the mesencephalon. Then in the area of the myelencephalon, general somatic efferent fibers are carried through hypoglossal nerve to striated muscle. These are the general fibers, motor fibers to the pharynx. Then the special visceral efferent fibers, these are the striated muscles of pharynx, cranial nerve 9 and 10. Then the general visceral efferent fibers to the smooth muscles, cranial nerve 9 and 10. General visceral efferent fiber, these are sensory fibers to the visceral structure. Special efferent fibers are carried through the cranial nerve 7 and 9 for the taste. General somatic efferent fibers carried through 5, 7, and 9, carrying the pain, touch, and temperature sensation from area of head and neck. And then in the area of the mesencephalon, we have general visceral efferent fibers carried in the cranial nerve 3rd nuclei, carrying parasympathetic fibers to the eye. This is another diagram showing you the different nuclei in the adult brain. The red color shows the motor nuclei and the blue color shows the sensory nuclei. So first we are looking at this. This is the red nucleus and this is the accessory oculomotor nerve nucleus, the Edinger-West wall, along with the motor nucleus of the accessory nerve. Then is the trochlear nerve nucleus, and then is the nucleus of the motor nucleus of the trigeminal nerve. Here is the nucleus of the abducent nerve, the facial nerve, then the rostral superior and inferior salivatory nuclei are present over here. Then is the nucleus ambiguous and the accessory nerve nucleus, and here you can see this one is the dorsal nucleus of the vagus nerve and the hypoglossal nerve. Nucleus of the hypoglossal nerve. So these are the different nuclei which are present in the adult brain, and now we are looking at the sensory nucleus. So this blue color shows the sensory nucleus. This is the mesencephalic part of the nucleus of the trigeminal nerve. This is the spine. This is the pontine part of the nucleus of the trigeminal nerve, and the spinal part of the spinal trigeminal nerve nucleus. So these are different nuclei which are present in adult brain, their cross section, and they are derived from the alar and the basal plates. So mesencephalon, the cerebellum develops from the thickening of the dorsal parts of the alar plates. Neuroblast in intermediate zone of the alar plate migrate and differentiate into neurons of the cerebellar cortex. Cells from the alar plate give rise to dentate nucleus. This is the largest nucleus. The pontine nucleus, the cochlear and vestibular nuclei, and sensory nuclei of the trigeminal nerve. And bands of the nerve fiber crosses the median plane and form the bulky region, that is the bones. So So this is again the same diagram. Here you can see this is the upper part. In the here the lateral walls roof plate is thinned out, and in which you can see the perimeter and ependymal roof. And this is area of developing cerebellum. This is the cavity of the fourth ventricle. These are the pontine nuclei, and these are the basal and the alar plates which are forming different nuclei. And this section is taken over here at this level, developing, and these are the developing bones and the medulla. So next is this diagram, and this is in this diagram you can see the developing cerebellum, in which different parts of cerebellum are forming. This is the primary fissure of cerebellum which is forming the anterior lobe, also called the paleo-cerebellum. This one is the posterior lobe, also called the neo-cerebellum, and the flocculo-nodular lobe forming the archicerebellum. So these are the different parts of the cerebellum. This is the cavity of the hindbrain called the fourth ventricle, and this is the choroid plexus or the tila choroida which will form the CSF. So choroid plexus is formed in the ependymal roof of the fourth ventricle, covered by biomatter externally. It actively proliferate to form tila choroida and invaginate into the ventricle through different foramina, and it forms the CSF. In the region of the midbrain, the neural canal that passes through midbrain narrows, and the cerebral aqueduct is the canal of the midbrain. It connects the third ventricle to the fourth ventricle. Neuroblasts migrate from the alar plate of the midbrain into tectum, that is forming the roof, and aggregate to form the superior and inferior colliculi. So these are the sensory nuclei which are concerned with visual and auditory reflexes. These are derived from the alar plates in the area of the midbrain. Neuroblasts from the basal plates give rise to neurons in the tegmentum, that is nucleus of the red nucleus, the third and fourth cranial nerve nuclei. The reticular formation and substantia nigra is also considered to be derived from the basal plate. The cerebral peduncles fiber from cerebrum passing through the midbrain, brainstem, and into the spinal cord. So this is the diagram. Here you can see this is the prosencephalon, the mesencephalon, and the hindbrain are the rhombencephalon, and section B is taken at this level. In this diagram you can see these are the primordia of the colliculi which are present over here. These are alar plates and basal plates. Substantia nigra is developing, and these are the crus cerebri. So these two sections are taken in the midbrain. E is at the level of the superior colliculus and D is at the level of the inferior colliculus. So in sections D you can see both the inferior colliculi, also the mesencephalic nucleus of the trigeminal nerve is there, the trochlear nerve nucleus which is somatic efferent over here. These are the decussation of the superior cerebellar peduncle and interpeduncular fossa, and this is the crus cerebri and substantia nigra which is developing. So this diagram is taken at the level of the superior colliculus, and in these two you can see superior colliculi which are differentiating, and also at this level you can see mesencephalic nucleus of the trigeminal nerve, also the oculomotor nucleus. So please differentiate. At the level of inferior colliculi you will see the trochlear nerve nucleus, and at the level of the superior colliculi you can see the oculomotor nerve nucleus. Also you can see red nucleus at the level of the superior colliculi, while at the level of inferior colliculi you will see the decussation of superior cerebellar peduncle. So please dear students remember that these points will also help you to memorize the sections of the midbrain at the level of the superior and inferior colliculi, both in the neuroanatomy as well. So here I complete my today's class, a little bit lengthy and difficult class. The references are Langman's Medical Embryology, Developing Human by the Keith Elmore, and the Gray's Anatomy by the Susan Stranding, 4th edition. Thank you all dear students for your attention for today's class, and we will meet in the next class where we will do development of the forebrain and remaining part of the CNS. Till then, Allah Hafiz.